Hi, my name is Katrina Hatch, and today we're going to be talking about genetics, but most importantly, we're actually going to be talking about DNA replication, translation, and transcription. Within replication, there are actually three parts. So I'm going to try and break it down into a way that's better understood. So the first part of replication is called initiation. And initiation is where you're going to find the origin on the circulated DNA, because right now we're mainly focusing on prokaryotes. And you're going to find the origin, and when you find the origin, this is where the second part comes in. And the second part is called elongation. So within DNA elongation, there is a lot of vocabulary. So we're going to try and start with the basics first and try and move from there. First off, we have this green strand, which is going to be our template strand. And then we're going to have the blue strand, which is the non-template strand. The template strand is going to provide the directions for the strand that we're going to try and create, basically. So when elongation begins, what's going to happen is you're going to have a replication fork at the origin start to form. And the replication fork goes like this, and it's just going to start being a hole, and it's going to go down, and it's going to unzip. And what unzips it is called helicase. And let's use this little cotton ball right here as our little helicase. So it's going to go down, it's going to unwind, and then you're going to have another cotton ball down over here that is going to be basically relieving the stress it's going to be forming because you're unwinding down here. And this is called DNA gyrase. So then you're going to be over here at your replication fork, and you're going to have the leading, or it's going to divide into two strands, and you're going to have your leading strand and your lagging strand. And I know I called these the template and non-template strands, but for this particular example, this is going to end up being our template strand. And so here we're going to have the template strand is going to form the leading strand, and a leading strand is actually a continuous strand. And what's going to happen is you're going to have little primers. Where's my primer? You're going to have primers that are going to be set down. And I don't have any extras, so we're going to put it on our leading strand. And so with this primer, what's going to happen, this actually should go on the inside, is it's going to be laid down by primase, and that's an RNA primer. And then it's going to lay down, you're going to have DNA polymerase. It's going to come in, and it's going to start laying down other nucleotides to kind of correspond with these. And these are going to match up, you know, A's with T's, C's with G's, just like it would with DNA. And DNA replicates in the 5 to 3 direction. And so the leading strand is going to be 5 to 3 because this green strand is actually running 3 to 5. And so it can replicate continuously in the 5 to 3 direction. Now this strand, however, is not going to work like that. And so what's going to happen is, I'm going to take this primer right here. This primer is going to be added in many areas. So there's going to be like a primer here, there's going to be a primer here, and it's just going to keep going down. And then what's going to happen is at these primers, you're going to have polymerase also that's going to come in, and it's going to lay down these little fragments. And these are going to go in the 5 to 3 direction, because this blue strand goes 5 to 3. So you're going to have to replicate this direction. And it's the opposite direction of the helicase. And so that's why you have to make it fragments, because it's going to have to, you're going to have one here, and it's going to have to go backwards and add this one. And it's just getting complicated now. So that's what's going to happen here with the leading strand. So this is going to be complete. And then you're going to have the lagging strand. And these little fragments right here are called Okazaki fragments. And what's going to happen is with these Okazaki fragments, you're going to have, where is it? You are going to have this thing called ligase come in. And the ligase is going to attach these fragments together, just the gold fragments, but the blue one kind of got in. So the little tape is DNA ligase. But before this ligase actually acts, because I didn't do it over here, you're going to have this primer. And with this primer, this is an RNA primer, and you can't attach an RNA primer to a DNA strand. So what you're going to do is you're going to have DNA polymerase III come in. We're just going to use another cotton ball, and it's going to magically transform it, and it's going to change the RNA primer into a complementary DNA bases. And so you're going to have like a little gold strand here, and then you're going to use the ligase that I used over here to put the whole strand back together, and it's going to look like one long continuous chain like this one. So this process is actually going to continue. So you can have your little helicase over here, and it's going to continue down the entire strand of DNA. And so, I mean, like, it's all going to unwind because of the helicase, and then you have the gyrase over here kind of fixing the torsional strain. And so all of this is going to be, we're just going to stretch this out because it'll be easier. So this blue strand is going to be, it's going to be connected by all the little fragments. It's going to have all its fragments, and it's not working right now. And then you're going to have this green strand, and it's just going to continue replicating, and it's going to have continuous replication, or continuous laying down. So it's going to have again, this is the leading strand, so it'll be, you won't have to connect any fragments. And then what happens is you get to the end of the line, and when you get to the end of the line, you're going to have the third part of replication called termination. And in order to actually get to the end of the line, you need a termination sequence. And this is a sequence of data that basically tells the polymerase and all the other molecules that, stop replicating, we don't need it anymore. This protein is going to come in, and it's going to stop replication. And so what it's going to do is it's going to bind to the DNA site, and it's going to bind to the site called the DNA replication termination site, and it's a binding protein. So it's going to bind here, and it's going to stop replication. And then at this point, it can move on. It can be its own strand. So now it's a double-stranded DNA, and it can now move on into the next step of DNA processing, and it will be called transcription. All right, so now we're going to move on to transcription. And transcription is the second thing I said we were going to talk about. And it's the process by which DNA is converted into mRNA. And in order for this to actually happen, in order to get the final mRNA strand that you want, it has to go through a lot of processes. So what we're going to do is we're going to start off with these two strands. And we're going to do mRNA formation. With mRNA formation, it's very similar to DNA replication, which we just went over. But the only difference is that only one of these two strands present are actually going to be transcribed. So what's going to happen is one strand is going to hold the gene, and that's going to be called the sense strand. And then the other strand, so we're going to say that the gene is on the green, because that kind of rhymes. And then this gold strand over here is going to be the antisense strand. And that's the complementary strand, and it doesn't actually hold the gene that we're looking for. So the weird thing about the mRNA formation is that although this green strand holds the gene that we want, we're actually going to transcribe the antisense strand, so the one without the gene, so that way we can make a copy of it and produce the gene that we are looking for. So we're going to remove the sense strand, and we're going to start with transcription. So transcription is similar to DNA replication. So what's going to happen is, the primers are going to be laid down, and then you're going to have the base pairing come on, and so you're going to have these. So these are it's all similar genes, or it's all, A, T, C's, and G's. So it's going to line up like this. So yeah, you're going to remove the primer, and then, the polymerase, the ligase, and all that other stuff, it's all going to come in. And it's going to make this all one strand. So this is actually all one strand. I just have them looking at different, and we're going to talk about that in just a second. So then what's going to happen is we're going to move on to the second part. And the second part that's important with transcription is called mRNA editing. So what's going to happen is our mRNA editing is going to take place. And so we're going to splice out these, we're going to cut them out. So we can use these little scissors over here, right? We're going to cut out these introns because they're going to stay inside the cell. And we're going to leave exons. So these are exons. We're going to take out the intron right here, and we're going to leave that exon right there. So what's going to happen is these are then going to move together, and then we can actually remove this strand because we don't need this anymore because we have our mRNA strand. We don't need the template. And so these are going to come together and form the mRNA strand. And what I actually did for this example is I just took out all the introns, but there's actually this thing called alternative splicing. And that basically says that, you know, I could leave one intron, and basically you could potentially form multiple different types of mRNA strands. And this is why you can have one gene, and end up with multiple, like thousands of proteins. And so it really only takes one gene. It just matters on how you splice them and how you get them out of the cell. Then what's going to happen is you're going to go, this little strand right here is going to go through mRNA processing. And what happens there is that you're going to have the 5' cap. It's a guanosine cap. And then, you're going to have the 3' tail. It's a poly A tail. And this has a bunch of like adenosines on it. And these two things together are the only reason that that this strand can actually leave the nucleus, because this is all occurring within the nucleus right now, because that's where you can find the DNA. So then what's going to happen is this is going to move outside the nucleus, so you know you're going to transport it. And then now we're going to pretend we're in the cytosol. So now that we're in the cytosol, cytosol is actually where translation occurs. This is the third step that we were talking about. And this is the process where this mRNA strand is actually going to be turned into a protein. And so what you're going to do is this mRNA, these are going to be these pairs of 3, I guess they're not really pairs, groups of 3. These are actually called codons, and these are located on the mRNA strand, like I said. And what you're going to have is you're going to have little tRNA molecules come in. And so here's a little tRNA. And it's going to have a little, we're going to call this an amino acid, even though it's a really big amino acid. And this blue code is 3, or has 3 molecules. And what it's going to do is it's going to match to these base pairs right here. So these 3 base pairs match with this one. And these 3 base pairs are called the anticodon. And in order for this first thing to start, you need the start codon. And the start codon is always AUG. And so this is going to bind over here, and it's going to happen, and I can't really show you properly, but we're going to call this the ribosome. And you have the large ribosomal unit, and you have the small ribosomal unit. And so this is this little guy over here. This little tRNA is going to come in this first slot, and so this is actually going to be divided into 3 parts. You're going to have the A site, P site, and E site. The A site is where this first molecule is going to come in. Actually, no, for the first time, it's going to come in the P region, and it's going to bind to the start codon, and it's going to drop off its first amino acid. And this first amino acid is always methionine, because it has to match with the AUG. But before it can leave, you have to have a second tRNA molecule come in, and it's going to have its other anticodon, so we're going to use this one again, and it's going to come in the E A site. So when it comes in, it's going to drop off its own amino acid. So you have this guy in the P site right here, and this guy in the A site, and they each have their own little amino acids. And So when it comes in, it's going to come in, and then this guy is going to shift, and when it shifts, it's going to pull this, or this guy's going to shift, and it's going to push this guy out. And when it pushes this guy out, these two amino acids are going to bind together. This little tRNA is free to do its own little business, and it's going to leave. And these are bound, and it's going to move from the, after it's moved from the A site and the P site where they're bound, it's going to be pushed out by another tRNA molecule. It's going to come in and bind its anticodon. And remember, this is moving down every 3. And so, you know, every 3 it's going to be a different sequence, and it's going to signal for a different amino acid or a different tRNA that holds a different amino acid. The cell has made, or has used this specific mechanism to help save space. And so what it does is it has this, it goes through this thing called the wobble effect. And basically what that means is on the tRNA, you're going to have three little different base pairs. And what that means is the third one, for certain amino acids, it can actually be, you know, it doesn't always matter what this one is. And so there are a few, like leucine, where it has 6 different types of tRNA molecules that signal for one amino acid. Then after all these, after you've gone down this whole strand here, and you've connected all these different amino acids, you're going to have this thing, and it's going to be a protein. And so this is going to be our protein right here. But in order to finish and let this go off on its own, what you're going to need is, you're going to need a stop codon. And there are 3 different stop codons. There's UAA, UAG, and UGA. And these are the only 3 stop codons. And basically what happens is when you run across, when the tRNA runs across a stop codon, that basically, there's no amino acid that is attached to a stop codon. And so what's going to happen is when it comes in, it is going to, you know, come in the A site, it's going to push it, it's going to push over the other tRNAs, and then when it leaves, it's going to detach this protein because it doesn't have anything else to bind to. And so now it's going to be its own little free-floating protein. And this tRNA molecule is going to do its thing, and then this ribosomal complex is going to leave, it's going to dissociate, and then this mRNA is going to go back inside the nucleus, and it's going to be degraded. And then those base pairs can be used to make even more mRNA molecules later.